And so now, without further ado, let's begin with the lectures. Please bring your hands together for a warm welcome and join me in welcoming our first speaker, Professor Jerry R. Mendell.
Thank you for this great honor, and to speak halfway around the world has just a lasting impression on me. I'm going to talk about how we really have changed the lives of children and babies with neuromuscular disease. And this work had never been done before. I want to emphasize that all that I talk about today will never have been invested by me in any way, so there are really no disclosures. I want to review the products that have been approved for neuromuscular disease that I have had significant influence in achieving. The first thing is a rather ordinary one in many ways, and that's corticosteroids. All of you are familiar with this. But back in 1989, when we published this in the New England Journal, there was no treatment for Duchenne muscular dystrophy. And I have this here not because it was such a great achievement in terms of corticosteroids, but rather because how it has influenced us in gene therapy, maybe much to our surprise. But corticosteroids do change the natural history of the disease. It improves muscle strength for 3 to 6 months, and then there is a gradual decline, but it changes the rate of decline. And it has its usual problems with side effects, as you're all familiar with. But now it is the standard of care for Duchenne muscular dystrophy patients. Other steroids have been introduced since that time, and one thing I like to comment on is that since we published that in 1989, there have been 59 publications that have confirmed the findings, and that is one that makes a clinical scientist, a translational scientist, feel that their work has been worthwhile. The other steroid that often is substituted for Duchenne is deflazacort. It's actually a manufactured steroid, but has similar findings and perhaps fewer side effects, but has its one downside: it causes cataracts. And all these steroids have changed the ambulation time for patients with muscular dystrophy by adding 2 or 3 years. That doesn't sound like a lot, but it is in the life of a child. The other one that has recently been introduced, and I thought I would comment on this, this is not my own work, but important work, with vamorolone, which is a steroid that is considered to be dissociative. It has influence on the NF-kappa B inhibitor and decreases inflammation, but avoids many of the side effects, the dissociative effect, by not linking to the steroid hormone linkage in the nucleus of the muscle fiber. So let's talk about some serious stuff now, but I wanted to introduce the idea of where steroids fit. And I'm going to talk about the FDA-approved gene therapy projects. In order to achieve this is a monumental amount of work that goes into this. And I have been devoted to this for the past 50 years, making a difference in the lives of these children. I'll talk about when I first saw my first patient with muscular dystrophy. But let's just look at when I got into the gene therapy world in 1999, did my first clinical trial. The field looked rather miserable. It was highly criticized. Gene therapy was said to be short-lived because the promoter for the transgene would be automatically shut off. There would be causes of problems in the liver because the gene would race to the liver upon infusion and cause inflammation. and there would be something called insertional mutagenesis, where the gene would land in the wrong place in the nucleus and in the genome and cause serious side effects. Unfortunately, all these things turned out to be true, and that's where being a translational clinical scientist has its advantages, because I deal in all my years with patients, and being the person who could translate from the lab to the clinic made a difference in terms of handling these side effects. And the viral capsid was a particular site of immunity that we had to deal with. So I want to just introduce the concept of the choice of virus for gene transfer. This was the result of actually an unfortunate event in the gene therapy world using adenovirus. Adenovirus would have been ideal to do gene transfer because it's very large. It's 200 times the size of adeno-associated virus that we use now. So it could hold large genes like the Duchenne muscular dystrophy gene. And that was a sad event. In fact, I was just about to launch our first trial using adenovirus when the unfortunate death of Jesse Gelsinger occurred. And Jesse Gelsinger was a patient who had ornithine transcarbamylase deficiency and received the gene, and 4 days later had serious side effects. Perhaps this wouldn't shock us so much now, but in 1999 when this happened, when gene therapy was trying to make a foothold, it was considered to be a major event. And of course, the death is a major event at any time, but this was in a new field, and it predicted, it was the result of the result of the prediction. The prediction did say that things horrible would happen with gene therapy, and this is one example. So the other one was using retrovirus. Retrovirus is also a very large virus. It would hold most of the single gene monogenic gene disorders, and in X-linked severe combined immunodeficiency disease, it was used in the children who had that, and 5 of the 20 developed T-cell leukemia because the gene inserted at the wrong site. And retroviruses take the gene, bring it into the nucleus, insert it into the genome. That has great advantages in many ways, because once it's locked into the genome, then it can't go anywhere. But the downside is, when that happens, the investigators and the children have no choice either, and so then you have to treat the adverse events that occur as a result of that. So that left us with what we turned to was adeno-associated virus. This is a virus that's very small. It's 200 times smaller than adenovirus. It gets its name because it requires a helper virus to propagate, and adenovirus is one of the helpers, and so is retrovirus. So, but it has its name. This name actually, I would say, is not a blessing in the sense that people get confused about it, but it's a single-strand DNA virus, and it has no human disease. Well, in some ways that's almost a joke, because we've dealt with this in many ways that I'll discuss. It has multiple serotypes, over 100. Serotype is just a change in the amino acid composition of the surrounding envelope of the virus, the so-called capsid of the virus. And the two I'm going to talk about are AAV9 and RH74. And both of these had not been introduced in clinical trials until the ones I'm going to tell you about, nor had they been used in anything like the doses that we used. And the strength was it was non-integrating. Now remember that HIV, or the retrovirus, integrates. Here we had a virus that didn't integrate, and that was considered a weakness initially, but then after the Jesse Gelsinger affair, it was considered to be a strength. And you'll see how and when we first initiated gene therapy, we used that to an advantage to bypass FDA concerns about even starting over after Jesse Gelsinger. Its weakness is it has limiting packaging capacity, and you can see that here. I just have a little drawing on the side there that shows the change in relative size of adeno-associated to the other two bigger viruses. So let's talk about this fact that we went into gene therapy using adeno-associated virus under the assumption that there was low immunogenicity. What a marvelous little guy this was, could go anywhere, non-integrating, no immunogenicity, but that was deserving of the quotation marks I have it in. Because one of the things we learned very early was that that this virus was immunogenic, and not surprising, but naively entering that. And I have this picture up here because the first patient that I ever did with was in gene therapy was limb-girdle muscular dystrophy. I'm not sure how much that means to you, but we can elaborate on that to some extent. But I wanted to emphasize that. And the reason we chose that is this is one of the sarcoglycans. There were 4 sarcoglycans that were initially associated with limb-girdle dystrophy. Now we have 30 limb-girdle dystrophies that are identified by different genetic sequences. And this was an LGMD2D trial. We did the first one in 1999 where we did first 2 patients, but then that was the same year that Jesse Gelsinger had his disaster, and the FDA would not let us complete that trial. It was also the time that I went from really what we call main campus in Ohio to the Children's Hospital exclusively. And at the Children's Hospital, I was able to build a gene therapy program that really was one of the first basically in the world. And we did our first, after the initial trial starting in 1999, we worked toward building another trial that we initiated in 2006. And this is a typical picture of a limb-girdle dystrophy patient on the left. They have proximal weakness. They have loss of ambulation, usually the sarcoglycans in teenage years, and are much like the more severe disease, but a little bit milder. And one of the strategies that I used for treating this disease and going to the FDA, and this is where learning about gene therapy becomes very important. It's more than a mouse study, going to put a gene in a mouse in the lab. That is absolutely necessary, but then to convince the FDA that you've got a safe product that you can bring to the clinic is a different story. And I decided at that time that the best thing we could do was, there's a tiny little foot muscle that all of us have. It's called the extensor digitorum brevis. And I could inject that muscle, and then if there was a problem, remove it quickly so that the patient wouldn't have any problem. And as you can see here, that's actually me injecting the gene and using ultrasound to monitor where the gene is so that it avoided any blood vessels. And then we had electrodiagnostic evidence that we were in the right place. And this patient is still, he's ready and willing to have another gene therapy, but this was rather bold on his part too. And he's now, I think, in his 40s and ready to go again. But in that trial, we had very good gene expression. We did 6 patients, and on your left you'll see that we had robust gene expression in 5 of the 6 patients. This is where the idea of immunity comes into play. But in one of the patients we had no expression, and that was a puzzle in a sense, probably not too sophisticated a puzzle, but we were told there was no immunogenicity, and yet here you can see at day 2 we had a rather high peak of virus immunity, T-cell immunity that was exhibited by this graph that you can see, and then so we knew that we don't ordinarily see an immune response within the first couple of days. It's usually the immune responses are maximum at the 2 to 3 week point. So we knew that we had immunity. Now, not only is it unusual to have immunity, but we have what we call pre-existing immunity. And I show this because this is a major lesson that we learned in gene therapy. We learned that some patients will naturally acquire the immunity pre-existing to the gene therapy. And by pre-existing to the gene therapy, that means if you don't check the immune status of the patient to the specific virus you're going to use for gene transfer, you can end up with some pretty serious side effects, and I'll show you that in the next couple of slides. So that was lesson one that turned out to be very significant, and this was way back in 2006. And just to show you, illustrate about pre-existing immunity, here you see that patients 1 through 5 in the middle column have pre-existing immunity that's less than a dilution of 1 to 50, and after day 7 it went up to 1 to 3,200, which is not a surprise. But look at the patient number 6. By day 7, his immunity was over 100,000 dilution. So in a sense, what we know happens is that the antibodies that are circulating link to the virus and prevent uptake into the nucleus. So now we have established in our first gene therapy trial, we established the guidelines for all future gene therapy trials, which was very significant. And the other one that is really fascinating is what we found when we did our first trial with Duchenne dystrophy. This was a totally different kind of immune response, and here now we've got gradual. I don't want to talk too much exactly about how we advanced gene therapy, going from foot to limb to then become systemic, but just so to establish the idea of going in slow transition to really what makes a clinical difference. But here it is, I'm injecting our first Duchenne patient into the biceps muscle. Again, these are local intramuscular injections, but the amount of information we learned is overwhelming. And this was our first report of an immune response to the gene, not to the virus now. And this was published again in the New England Journal and had a profound effect and was the Science Achievement Award for that a year. And this was 2010. And really what happens is, what's the lesson that was learned here? This is actually the top cartoon shows the protein for the dystrophin protein that's a result of the gene. So it's a normal dystrophin protein, and it's 14, and the gene that delivers this protein is 14 kb. And we have on the bottom a picture of the parallel part of the gene that's compromised in order so that it will fit in AAV. And I'll show you a clearer indication of that. But this patient was deleted of exons 3 to 17, which is common in Duchenne dystrophy. Deletions are the most common mutations. And what we found when we injected the gene was that this child had developed antibodies and T cells that were generated from the area of lack of expression of the, so this was a novel new protein to this patient. And basically the message was, don't inject the gene into a patient who has a parallel area where the gene is missing, because the protein that's produced will cause an immune response. It's no great science, but people didn't believe that transgenes caused immune responses. And this is really more shows the science of the of the gene. On top is the Duchenne gene with all the exons shown, and there's 79 exons. And in order to get the gene into AAV, we have to remove more than 50% of the gene, and we remove it from the central part of the gene, all the insignificant or less significant parts of the gene to express the protein. And here we go. That's the modified gene now that is missing 44 exons. And when we started in gene therapy, we were, and still are, a little bit farther ahead in the field than many, but we had competition from Genethon, Pfizer, and Solid, and other big pharma. We were small pharma, but this is big pharma who wanted to have really play in this field. And when we injected patient 3 to 17 deletion, that's where it overlaid in terms of the gene, and so that was missing, and now what you can see is that this part is missing here, and when the gene is expressed in that and the protein is expressed, we have a novel protein. Now let's look down here, because this was highly criticized. This would never happen, despite the fact that it was published in the New England Journal. We had all kinds of letters to the editor that this was kind of absurd science. And here we have 4 patients from different trials now, and that really strengthens the confirmation of this concept, not from our trial, but we did, again, another naive thing in one of the patients. We injected a patient who had a 3 to 43 mutation, that big red area. That's how much of his gene was missing. And we injected our gene, 1 to 17, into this area, and of course there was an immune response. And then here comes along Genethon with an 8 to 13 transgene and injects, or an 8 to 13 deletion, injects their gene, which is also 1 to 17, and Pfizer comes along and injects their gene. Now this got to be pretty serious. When we did it at first, we were fortunate, and I always say that somehow I have been the most fortunate translational investigator in this field. But these resulted in more serious side effects. Now we had inflammatory myopathy, myocarditis, thrombotic microangiopathy, and Pfizer had to deal with deaths of patients. So again, the Jesse Gelsinger mark of severity manifests again, and really re-emphasize, don't inject the gene into an area that the patient's deleted, and that goes for every gene in the gene therapy world. So now let's move on to how we applied these principles to gene therapy. And we chose SMA as one of the starting points because, as I explained, AAV is a very small gene, and SMA is only 2 kb as opposed to Duchenne muscular dystrophy, which is 14 kb. So we wanted to put in something that would fit. But the problem in genetics of SMA, it was really very complex. SMA stands for spinal muscular atrophy, as I should comment on. And this is a disease, I'll show you on the next slide, that results in death, uniformly death by 2 years of age. No other end of the story. And Judith Melke, who is a brilliant investigator, developed and understood what was going on here. And there are 2 paralog genes, SMN1 and SMN2. But the problem is in spinal muscular atrophy, SMN1 doesn't work. It has a mutation and doesn't work from the very beginning. And SMN2, which seems like it would be compensatory, also has a mutation. It has a C to T mutation, as you can see I have illustrated up above, and that only allows, it's a missense mutation, so a small amount of protein can be produced, but we're talking about 5 to 10%. It won't rescue these kids from natural history. There are milder forms when there are multiple SMN copies, and that will suffice for extending the life of these children. But what we did is not so hard to think about. We put SMN into AAV and injected it into patients, and now we could produce full-length protein and basically save the child's life. And this was there was another piece of information here that was important, and that was that how could we? There were those naysayers out there that said that the gene AAV would never cross into the blood-brain barrier, but there was a young investigator and a collaborator of mine who worked at Children's Hospital who had shown that genes could cross the blood-brain barrier in a model that was developed for polio, and this was AAV9, and AAV9 was unique in that it could cross the blood-brain barrier. So that's what we loaded. We loaded the SMN gene to AAV9 and changed the story. But just so you understand the seriousness of this disease, children are born sometimes almost normal at birth, but within the first few weeks or the first 3 months, they develop severe hypotonia, bulbar muscle weakness, where they can't swallow, difficulty breathing, and this is the natural history curve. 50% are gone by 10, 11 months of age, and all are gone virtually all are gone by 20 months of age, and all are definitely gone by 2 years of age. And this is on a clinical sense, this is an easy model to construct a gene therapy trial for, because you can say, if children are going to live beyond 2 years of age, my God, you have a winner here. And that was our model for the planned program for the outcome of the clinical trial. And what we did here was develop, and the FDA likes to see this, although it's not so good for patients and their providers, is that we had to have a dose escalation trial. So we knew these were safe doses, actually at 6.7 e to the 14th genomes, vector genomes per kilogram, and we injected the first 3 patients at this lower dose with a plan to go up even higher, but the highest dose of virus that had ever been given in a clinical trial. We knew it was safe from our preclinical toxicology studies at 2e to the 14th vector genomes per kilo, and we planned another 12 patients, the highest dose now ever given. And this is what happened. We gave the first dose in almost a nervous breakdown. We had the big red line there as a T cell response within the first 2 to 3 weeks, and liver enzymes now. We thought we were smart enough to avoid liver injury, but we had liver enzyme elevations in the 13, 14, and 1500s, and that was very frightening. So I went back to the FDA and said, hello, we have excellent data in children for using prednisone. Can we use it now if we give it 1 day prior to the gene therapy to blunt the immune response and see if we can get an effect? And yes, we can. As you can see here now, we have almost no liver enzyme elevations and a minimal T cell response. And so this now becomes the precedent for gene therapy trials, and that again why I linked prednisone in these children with neuromuscular disease. And here's a picture of a longitudinal picture on this graph. The first 3 patients are the first 3 lines on top, and given it at a low dose. And after day 1, what you see here is prednisone is administered, and now we could give the higher dose, 2e to the 14th. And the most important thing is that every patient achieved the primary outcome. There were no deaths before 24 months, before 8 months of age, and no deaths by 2 years of age. And we had even more profound results when we looked at patients longitudinally, and I'll show you that. I show this picture because, well, since this was the highest dose of virus ever given, what would happen? What would the immune system do seeing this? There'd be naval immune responses that didn't require pre-existing immunity. But the remarkable thing was there was no reaction, and that's pretty much true for gene therapy across the board. Gene therapy is very well tolerated, even despite these very high doses, at least initially, and this is especially true in babies. The most common response that we see in children is they have vomiting, and this will be 60% of the patients by day 3. This slide also shows an important point, is that early treatment is the key. And for those of you I met this morning who are working on newborn screening, what you're doing is contributing to the potential for gene therapy. And here we did first 2 patients that had been diagnosed by pre-existing gene because they had siblings that were involved. And those 2 that we treated within the first 4 weeks had virtually returned to normal almost within a matter of weeks. And if we gave the dose that was the highest dose, the earliest time point. If we gave a lower dose, even at an early time point, we could achieve significant effects, as you can see in this graph. But if we give the dose at a very late time point, especially a low dose, we achieve almost no effect. And that's true for almost every disease. And this is a model I think you should think of in terms of developing your own gene therapy program. So the other question is that still there are negative naysayers out there, is how long will the efficacy of gene therapy persist? And this is an example in this first trial. This is our follow-up data on the patients who had achieved high dose. We had 2 that walked almost immediately, 2 that developed were able to stand after the first 2 years. And then all were able to sit this is a graph at the 7-year post gene therapy time point. And now if you look over on the right, we have a gorgeous little girl here who's able to sit and will take your heart away. And then the boy who's now 10 years old, who's standing and having basically a normal life. He was the earliest one that we treated in this clinical trial. And now this product, this is some other examples here. We had by 6 weeks of age, by 6 weeks post gene therapy, we had babies who could sit up and hold their head and drink from a bottle, or an indication on the neurologic part of a well baby is one who can lift his head by very early age. And this is 27 days. This baby was dosed at 27 days and 4 months post gene therapy. He had normal head control. And this is body control. That was now 6 months post gene therapy, and good control in the upper right corner with extension of the legs and good trunk control. But this is my favorite one, where we have a boy now who's able to climb up a wall in a local park. These were videos that were sent by the parents, and this is my favorite one down here on the lower left. I call him the American Ninja Warrior, and and then to add to that, here he is ready to go off to work carrying his briefcase and going up the stairs. So I think this is pretty good confirmation, and these kids now are 10 years, this one's 10 years old and has not lost any significant functional outcomes. So if you get it early, you can really reverse the disease. And shortly after we had FDA approval in the U.S., virtually every state, they're state controlled, approved newborn screening, and now that's done. And they don't need people like me anymore. We've translated it not only in the science world, but now into commercialization and into delivery by providers in the community. So this is a very gratifying response, and one that this is really the first systemic gene therapy. If I didn't emphasize it, now this is IV gene therapy. We went from IM to, I didn't talk about single limb gene therapy, we did that, then we went to systemic gene therapy with these very high doses. So what we used there was, well, what we used for Duchenne muscular dystrophy. And these are taking the principles we learned over 20 years and now applied them to Duchenne. And the first patient we did with Duchenne was in 2018. In January of 2018, in fact, it was on my mother's birthday, which must have had some significance in helping us move this along. And this was, the reason I like to show this slide, because the first Duchenne patient, this is the patient I have as the first Duchenne patient I ever saw. That was back in 1969, so you can extrapolate somewhere to my age and probably figure I shouldn't be here anymore, but I'm still going. And the gene therapy is the reason, because it's such a stimulating part of our life, and this 7-year-old now is long gone. But I committed, when we saw him in 1969, when I was at NIH for a postdoctoral fellowship, that we should make a commitment to change this disease. So what we learned in SMA, we applied to Duchenne. Now remember, we had to minimize this gene. And this was microdystrophin. It's less than half the size of the Duchenne gene. And it's called, because I had developed the gene with a postdoc of mine by the name of Louise Rodino-Claypack. And fortunately, the fellows I've had have all outstripped me, and you should always have your children do better than the parent. And now the postdoc who worked with me here is now the head of Sarepta Therapeutics. So I'm very proud of that. And we started this the doses shouldn't be surprising. The dose is the same as SMA. And we used, the age we used was between 4 and 7. We actually, that was what we got permission from the FDA, but the three patients, four patients we injected were 4 to 6 years of age. And now we excluded patients 3 to 17, no surprise, because the gene expresses into 1 to 17 because of its compressed, because it's a microdystrophin, and we don't want to inject the gene into areas that patients have deletions, so we check that. Now, we developed a different virus here, and this gets to the point of, you know, a blessed existence, or being in the right place at the right time. And why is that? When I went to Children's Hospital, shortly before that a new virus had been, a new AAV serotype had been isolated. It's called RH74 from a rhesus monkey line, and this was one that had not been used in clinical trial. If you look now, it's used everywhere because of these successes, but nobody ever heard of RH74, and that was good for us because we had in a sense, we had removed the competition from this, and nobody knew how well it worked. And so why RH74? Again, it was isolated at Nationwide Children's Hospital. We knew it had broad distribution, went to all organs in the body, And this is the construct that we use, and I don't have time to really explain the full rationale for the construct on the bottom, which has 4 spectrum repeats, 1, 2, 3, and 4 you can see that are SR, and it has 3 hinges and so forth. And this gene was shown to be, this transgene was shown to be effective way back in 2002. And also there had been a patient who had a mutation similar to this who had Becker muscular dystrophy, which is a milder form, and had lived past 60 years of age. So it seemed like the ideal gene to use for this capacity, and it fit well into AAV. And just to give you an idea of what we do, or not spend too much time on it, but we carefully monitor the patients during gene delivery, blood pressure, EKG. These were done in the NICU. Well, here's the result after the first 60 days. In the first 60 days, you can see up on the top are the pre-biopsies, which have virtually no gene expression, and then we have percent positive fibers on in the bottom pictures, and you can see had very robust gene expression, more than 90%. And using Western blot, there I have a full length where the full length should be, but the microdystrophin is what expressed in these patients, and the microdystrophin worked very well. And another thing about the Duchenne gene and its complex, it's very complex for its linkage in the cell. It links to other proteins in the membrane. It links to the refined parts of the muscle fiber deeper down into the structural parts of the protein. But when you reassemble in this case the dystrophin gene, you reassemble much of the membrane proteins in the cell, and now you have a protected muscle fiber. And as I showed before, now the post-gene therapy, this is immediately post-gene therapy of the very first patient we did, and you couldn't have a happier child. We give the gene over about an hour to an hour and a half, slowly infuse it so it doesn't all go to the liver, and it's very well tolerated. What we have to do is entertain the child more than anything else during the time of administration because of its slow delivery. And here it is. Much as I showed you, this is 6 weeks. This is 6 months post-gene therapy, and running very well. This is going upstairs. We're using reciprocal stairs, no hands. That can never be done by a patient who has gene therapy, who has Duchenne dystrophy. And here you can see 2 days post-gene delivery, how he walks upstairs, and 60 days post-gene therapy practically runs up the stairs using reciprocal legs to get up the stairs. And then the question comes up every time I would give this talk: how long does gene therapy last? We know it can't last very well. You put this gene in, it gets into the nucleus, it isn't in the genome, how can it stay there? Well, this is a natural biologic phenomena. When it's a single gene, single DNA, it forms double-stranded DNA in the cell, and then expresses like any other chromosome or any other gene in the cell, and works very well. And this is 4 years later, and you couldn't do a patient you couldn't show anything that's worth more than that. And this patient is now 7 years of age and continues to do extremely well. And this just shows different outcome measures. The North Star is one that we use, and you can see after the first year, this is at the 4-year time point, the North Star gets back to normal and stays at that level during that time. The other outcome measures here are time to rise, where you have a shortened time to rise, a shortened time to get up from the stairs, a shortened time to do the 10-meter and 100-meter walk. So unequivocal long-term results. And now on June 22nd, 2023, so this is not hot off the press, but not that far behind either. We had approval for gene therapy. And this was approval for the 4 to 5-year-olds, and now, if you see in the upper right-hand corner, we've submitted, and June 11th is the indication, a year later now, whether the FDA will approve gene therapy for all Duchenne patients. 4 to 5-year-olds was my mark, and what I did at 4 to 5-year-olds when we were approved, I decided that after more than 50 years, I would retire my position at Children's Hospital, but join Sarepta as a senior advisor. I had worked with Sarepta on multiple clinical trials over the years, but I will emphasize without any evidence of commercialization on my part. Sometimes I regretted that, but that's irrelevant. But this has been a very gratifying experience in my life, and we're still making great progress. We have had our most recent publication now back to the limb-girdle, which I consider back to 1999. We started at limb-girdle. We just published our first limb-girdle clinical trial in beta-sarcoglycan, and we have lined up to do the beta-sarcoglycan phase 3 trial for full clinical approval and move on to other forms. I said there were over 30 forms of limb-girdle, and that's the next phase. And I want to thank all the patients who participated in these trials. I hope I didn't go too far over the time limit here. And these are people marked on top there with asterisk, Louise Rodino-Claypack. She and I worked closely together, and she is now the head of Sarepta. I have a very special person marked there, Samuel Zaidi. She was by my side when we did our first SMA trial, and I invited her as an awardee, a laureate for this experience, the King Faisal Award. I had permission to invite a special person with me, and I invited Sammy to come along, and very happy about that. And these are other people, and it's been a wonderful experience. Thank you very much.
All right. Good afternoon. My name is Howard Chang from Stanford University, and I'm really delighted to be here. It's a really special honor to be awarded the King Faisal Prize and also share the stage with Dr. Mendell. She told you an amazing story, and I'm going to tell you something complimentary about the roles of RNA in gene regulation. We're really living through an exciting time of RNA science. Over the last decade, we've seen expanding roles of different types of RNA. You may know that in 1961, the existence of messenger RNAs was recognized as a key intermediary in the information transfer between DNA and proteins. But over the last decades, we've found that there's many more types of RNAs, including long non-coding RNAs and circular RNAs and other types of RNAs that really are involved in gene regulation. That is deciding when and where genes turn on and off. And of course, over the last several years, RNA really became part of daily conversation over the pandemic because it also becomes part of life-saving medicine. And you may have noticed, the sharp-eyed members of the audience, that it was actually 60 years between the discovery of a messenger RNA and an mRNA vaccine therapeutic. And we certainly hope that the next phase of discoveries and translation would happen much faster. So I'm going to talk to you really about this arc from discovery to medicine in two topics. The first one about long non-coding RNAs, and the second one about more recently recognized types of RNAs called circular RNAs. And I want to then sort of center our first question about a very fundamental biological process, but also relates to a fundamental medical problem. Many of you may know that the human immune system is really highly biased And that is that 4 out of 5 patients with autoimmune diseases are women. That is an average. Autoimmune diseases affect many people around the world. In the US, more than 50 million Americans. And you can see that that number is an average. So for a disease, a quite common one like lupus, the ratio is 9-to-1 female to male. I'm a dermatologist, and these are some of the patients that I see. But this really reflects I think, a double-edged sword, that the immune system has differences when it comes to also the response to cancer. So female patients, for example, with melanoma, do much better to cancer immunotherapy, as well as infections. And when people think about this class of problems, let's think about why. So for decades, people thought about different hormones. So of course, men have more testosterone, and females have more estrogen. More recently, we and others are thinking about the fact that there's different X chromosome dosage, and that is because women have 2 X chromosomes. And indeed, there are rare individuals with an XXY genotype, so-called Klinefelter syndrome, that actually are phenotypically male, but have a female-level risk of autoimmune disease. So that really says that the X chromosome dosage really matters. What I'm going to tell you about is our work and highlighting that is a very special RNA that not just the hundreds of genes on the X chromosome, but just one RNA could perhaps have a very key role. And that RNA exists the master regulator of X chromosome inactivation. So let me explain that, remind you that, again, males have an X and a Y chromosome, whereas women have 2 X chromosomes. And the system to make the gene expression output equivalent between the 2 sexes is that there's a long non-coding RNA called exist that is made only from the 2nd X chromosome, one of the 2. And for that reason, it is female specific. It coats the chromosome from which it is expressed and silences gene expression in that chromosome, and therefore equalizing the output between XY and XX individuals. It's a very powerful epigenetic mechanism. Just 200 molecules can silence across the entire sort of chromosome. And it's actually also, of course, a paradigm of the types of gene regulation of many other long non-coding RNAs that we and others have found. And indeed, there are, as I mentioned in a moment, the silence is incomplete, and there are genes that will escape X inactivation that defines sex differences in biology and disease. And this picture is a beautiful calico cat. It's a girl, okay? And you know that because this reminds me to tell you that X inactivation, once it happens, it passes on to all daughter cells of that initial cell for the life of the individual. So these different patterns of segments we see on the skin, these are clones of cells that have gone through X inactivation and silenced different genes and passed on to all its daughter cells. So really is a lifelong influence. Every cell in a female's body expresses exist and is continuing its role in gene silencing. Okay. And so if we think about what is happening at the DNA level, I need to mention to you that a very powerful way to look at this problem is looking at chromatin. And that is the DNA protein complex from which genes are housed. So important to remember that in every human cell, 2 meters of DNA is packed into a 10-micron nucleus. And therefore, most of your DNA is highly compacted, except for the active DNA elements that are driving gene expression, such as promoters or enhancers, which Dr. Mendell also talked about. And so simply reading the active or accessible part of the genome tells you the software program that the cell is actually running. And so to find these active elements, some years ago, my lab, together with my colleague Will Greenleaf, developed this method called assay of transposase accessible chromatin, or ATAC-seq. And the idea is that we can take an enzyme for a bacteria, the transposase, that copies and pastes DNA. And we load up this enzyme with DNA adapters that we can use for sequencing. When we react this enzyme to human or eukaryotic chromatin, it can only copy and paste into open chromatin regions. And therefore, in a single step, you selectively and covalently tag the regions of interest so you can actually amplify and sequence them. So this method led to a pretty remarkable, a million-fold improvement in the sensitivity and 100-fold improvement in the speed of mapping regulatory elements, really leading this revolution in epigenomic discoveries. So this is what the data will look like. So here on the x-axis are the locations of genes. The y-axis indicates the level of accessibility. This is the prior gold standard, DNase hypersensitivity. It needed 10 million cells. The second row was the first version of ATAC-seq, used only 50,000 cells. And more recently, we achieved, actually a few years ago, single-cell ATAC-seq. You can see that the pattern is virtually identical. But now when you zoom in, this is now, every row is a single cell. There are 254 single cells here. And every position here is either 0, 1, or 2 reads, because it's a diploid genome. Okay, so in this way, this kind of analog data has turned into digital information. And this technology has now really been the cornerstone of all of the single-cell activities and single-cell epigenomics now driving the field of epigenetics. Now coming back to the study of X inactivation, we use this method to look at the X chromosome. You can see in a female cell, the active X chromosome has all these active regulatory elements, and they're almost completely silenced on the inactive X chromosome, except for the elements that escape X inactivation. The top picture is a version of ATAC where we have now turned accessibility into a color, and this is a method called ATAC-C. And I've paired that with RNA in situ hybridization for the exist RNA. You can see that everywhere exists, there's a black hole of accessibility. If I can just move it forward, okay, like that. If I go backwards, okay. You can see that really every place the red color is, there's a black hole, and actually, in fact, RNA is pulled in the entire inactive X chromosome into this condensed structure to the edge of the nucleus. And so it was long suspected that exist would work with protein partners. And so some years ago, we created an RNA-directed proteomic method, identified that exist, which is a 19 kilobase long RNA, interacts with 81 proteins, 10 through direct RNA-protein interactions, and the other ones through indirect protein-protein interactions. So this is a very long list of partners. How do we figure out what is going on? And so here we're aided by some prior genetics, which told us that a small region of exist called the A repeat was necessary for gene silencing. So in exist that's missing the A repeat, basically it would still be made, still accumulate and spread across the inactive X chromosome, but could not silence gene expression. And this is actually the work of Anton Woods. And so repeating the exist CHIRP mass spectrometry experiment, here on the x-axis is the full-length exist, and on the y-axis is the data, the peptide counts, for the A repeat deletion. You can see that everything is on a 45-degree diagonal, except 3 proteins whose protein count falls to 0 in the A repeat deletion, indicating that these 3 proteins must interact with the A repeat. This also taught us that the exist RNA is modular. Different parts of the RNA bind to different proteins, and there's a division of labor. And we can then focus on these 3 proteins and then quickly realize that this protein SPEN, which had previously never been associated with X inactivation, was a key driver of gene silencing and initiating X inactivation. To summarize this work, we learned that exist RNA is not only a guide, it basically marks the chromosome that must be silenced, but it's also a scaffold. The RNA assembles the set of enzymatic activities like a train organizes passengers. So this RNA would deliver all these enzymes at the same time to the right gene locus that to enact the biochemical activities at that position. So for example, when X inactivation happens, the action of SPEN first turns to the active regulatory elements and silences them by basically removing these active histone marks, H3K27 acetylation. A second module of proteins, called the polycomb proteins, polycomb repressive complex 1, then remembers this information through further histone modifications, enforcing gene silencing through subsequent cell divisions. We've learned a lot of interesting things about how X inactivation happens through subsequent studies. I told you about the role of SPEN. So when we knocked out SPEN, sure enough, X inactivation could not happen at all, so the X chromosome would not lose accessibility. But we learned that a number of elements on the autosomes, the other chromosomes, also gain accessibility. And actually, to our surprise, almost half of these were a class of endogenous retroviruses called ERV-K. So you can see that they all became active. And what we learned is that, in fact, I told you that this A repeat was very important. And the sequence of the A repeat is actually highly reminiscent of a virus-derived sequence called ERV-B5, the same family of RNA. So this is the secondary structure. So what we learned is that the normal job of SPEN is normally to survey the genome to make sure that these endogenous viruses do not escape and start expressing. If these viruses start to express, they would make an RNA. The cell would know that there's a problem, and then the SPEN will find the RNA and shut the locus back down again. And what this taught us is that X inactivation actually happened, evolved, by a process called viral mimicry. And that is that the exist gene exists in other species as a protein coding gene on the X chromosome. But what happens in humans and mammals is that this ancient virus jump into this proto-exist gene and amplified itself several times, so that that exists RNA starts able to attract SPEN, okay? So what we're saying then really is that the act of X inactivation is viral mimicry. Exists RNA is the device that this female cell uses to trick itself into thinking there's a raging viral infection going on this chromosome. And therefore we can mobilize a very powerful epigenetic machinery to come shut it down. And this then accomplishes the developmental goal of dosage compensation. This might be something like the original sin in the system, as I'll tell you then about the connection to autoimmunity. Okay. Now, I told you about the protein complexes in X inactivation, and that is something that happens very early in development in the embryonic stem cells. But I also told you that X inactivation persists throughout life. So more recently, we found that in adult somatic cells, for example, B cells, myeloid cells, that exists RNA continues to act and works with new and additional partners to shut down gene expression in each of these cell types. This is very important because, for example, in B cells, there's an X-linked gene called toll-like receptor 7. It's a sensor for exogenous viruses, including RNAs. And if it exists is not working, toll-like receptor 7 would escape. And basically, there would be biallelic, a double dose of expression. And this could actually drive the expansion of a class of cells called atypical B cells that I'll tell you more about in a moment. And this is actually very important in reaction to RNA ligands like COVID virus, but also self-RNA protein complexes. Okay. So coming back to this idea about autoimmunity, a few years ago, I really had a surprising revelation. And this happened, I think, in part because I'm a physician scientist. So you may know that in the US, doctors have to renew their specialty certification every 10 years. And I was in the middle of going through such a test, okay, and memorizing autoantibodies that are used to diagnose patients with autoimmune disease. So during the day, I was looking at these proteins, trying to figure out their function in biology, and at night, I was studying this list of autoantibodies and autoantigens. Then I realized that some of these genes were exactly the same. They're the same genes I was looking at. And then it turned out that a large number of the top enriched proteins are actually autoantigens. Okay, so here is, in this heat map, these are the associated proteins, and these are the autoantigens across a variety of different diseases. And this really led to a different idea that perhaps this associated RNA protein complex could be a female-specific source of autoantigens and therefore be a trigger. Some of you may know that there's actually precedent that large nucleic acid protein complexes could trigger immune response. There's a syndrome called CREST syndrome, which is autoantibodies directed against the centromere. And the idea is that large RNA protein complexes would have epitopes, which are features seen by the immune system, repeated multiple times in a polymer. And that format can cross-link immune receptors on T cells and B cells and activate them. So exists RNA fits the bill because it is itself a very long, large RNA protein complex. And furthermore, as I told you, the RNA associates on a whole chromosome, an even larger polymer. Okay? So if you think the centromere is big, how about a whole chromosome? So the idea then is that this complex would come out of maybe dead and dying cells and trigger female-level autoimmune disease. So we thought a while about how to test such an idea. And then the most convincing experiment we could think of is to make a male mouse express exist and see if we can confer female-level autoimmune disease. So if we can do that, that would mean you don't need female sex hormones. You don't even need a second X chromosome. Just this RNA could confer this female-level risk. So how do you do that? Well, if you express exist from any chromosome, it will shut down gene expression from that chromosome. It will be a cell-lethal event in a diploid male cell. So it took us a while to think about this, but I just told you that if you delete the A repeat, you can actually assemble 78 out of the 81 proteins and basically not influence gene expression. And so we created this mouse, which we can then turn on exist on command on chromosome 11, and then ask, would this male expression of exist confer autoimmunity? So on the right, let me just show you that this worked. We can now express exist throughout the body to a level seen in female cells in this exist male transgenic. And at the bottom, you can see that now these, if you, in the presence of doxycycline, which turns on the transgene, we can then see the foci of exist, so-called bar body, but now coded on chromosome 11 rather than the X chromosome. Okay? So now we need a model of autoimmune disease. And here we turn to a model called the pristane-induced model of lupus. Pristane is an organic chemical that when injected causes inflammation and tissue damage. And in the right genetic background, basically there's a much more severe female-like, females would have a lupus-like disease compared to males. And so there will be a lot of mice in these experiments, but just to keep things simple, all the figures are going to be color-coded, and we're just going to worry about a couple of the genotypes. So one is the positive control, that's the female mice treated with pristane, they get the disease. We have the negative control, which is the male mice, they get much less severe disease, and focus on the test case where we turn on exist, and we want to see if these mice get what kind of level of disease. So we are indeed sort of first notice that exist can induce autoantibodies, some but not all, in these males that express exist RNA. Okay, and this also led to complement activation. So some of the signs of immune dysregulation. But more importantly, we see that exist expression was enough to cause a female-level multi-organ disease, right? In kidney, liver, lung, and spleen. And some of the histology is shown here. So you can see that basically damage in the glomerulus or the liver, which is mimicked when male mice express exist. This is summarized in this total pathology score. You can see that 100% of the female individuals have this high level of the disease. None of the males have this severe disease. And with the expression of exist, now a majority of the individuals have this female level pathology. So we can come to the conclusion that exist can confer this female level autoimmune disease. Okay. Now, of course, if we're getting disease, we want to understand the molecular basis. And we can see that, in fact, exist is changing both the T cells and the B cells in these individuals. And this is now, again, using chromatin accessibility in CD4 positive T cells that we can identify patterns of accessibility that characterize male individuals versus females. If you start to express exist in males, we can see some of these patterns change over to the more female-like pattern. So they're effectively becoming, and we can actually tie it back to the fact that there are more naive CD4 T cells in the male. Basically, they become memory T cells in the females, and exist expression is able to actually induce that switch. And this is associated with elevated expression, for example, the toll-like receptor, another toll-like receptor gene that is approaching the female-like pattern. we went on to conduct very detailed single-cell analysis of both RNA and gene expression. And actually, to our delight, we found that the most expanded class of cells in these males expressed and exist is this atypical B cells. And this is the marker called ZEP2 that characterizes atypical B cells. You can see that they're high in females, but also high in males expressing the exist transgene. Now, why the focus on atypical B cells? So this class of cells gets its name because it is activated by a different signal. So the standard B cells are activated by immune receptor ligation and something called CD40. Atypical B cells are activated instead of CD40 by toll-like receptor signaling, which I told you about is an excellent gene receptor for RNA. All right, and so we believe that the atypical B cells may actually be directly sensing the exist RNA. And in fact, this is something we did not know at the time, that atypical B cells actually preferentially accumulate in aged animals, but especially aged females. So again, this female bias that connects very nicely with our hypothesis. Okay. I want to come back to think about patients. And this is a very sort of unexpected part of the journey, and that is that we thought, okay, this is great. We see this pattern. So should we see basically maybe antibodies against exist in our patients and also studies of autoimmunity? And so many of you may have heard about anti-nuclear antibody. Actually, let me see a raise of hands. How many here have heard of used ANA? Please raise your hand. Any medical students? Okay. This is the standard screening test for basically really working up autoimmune disease, and you basically take blood from patients, you put it on basically a substrate, and you see what structures light up. Okay, the first ANA was done in 1948, and but then as I was studying this, I realized that in 1975 a standard was chosen, and for whatever reason, it was a male cell line that was chosen. And so basically in the subsequent decades, right, so basically think about this, millions of tests were done on our patient. I told you that the majority of our patients are female, but this test was done in such a way that we would never see any autoantibodies to exist because male cells don't express exist. So in fact, there's a problem in that this cell line subsequently got contaminated with HeLa cells, different problem. But if we want to study sex bias in medicine, and we're not sure about the sex of the subject that we're studying, we can't make any solid conclusions. So here we are in 2024. What are we going to do about it? So to that end, we created really then a protein array comprised of all the exist-associated proteins. And these are antibody fragments, approximately 80 amino acids in length, so we can capture conformational and linear epitopes. And then we collaborate with clinical colleagues to examine blood samples from patients with a number of autoimmune diseases. You can see then on this volcano plot on the right then that indeed we see a number of elevated autoantibodies against the exist RNA protein complex in patients, really connecting this work that I told you about in mouse models to human patients. These patterns of autoantibodies can actually distinguish different types of autoimmune diseases, including scleroderma, lupus, dermatomyositis, and this may create then new opportunities for diagnosis. And finally, what I've shown you is a correlative experiment that, okay, so humans have these autoantibodies, but how do we really know that this is because of this RNA exist? And so here we come back to our mouse model. And here in this heat map, this is looking at the levels of autoantibodies. Every column is an individual, every row is a different protein, and the colors indicate the level of reactivity to that protein. The column on the left, very left here, in the orange, these are the autoantibodies in human lupus patients. And then the patterns on the right here is what is seen in the mouse model. So what I want to point out to you is that indeed female individuals have much higher levels of autoantibodies compared to males. But if we express exist RNA, we can now confer female level exist autoreactivity to a pattern that's indistinguishable to that seen in females, summarized on the right here. So this leads to the conclusion that exist RNA induces autoantibodies to its own RNA protein complex. Let me be clear that every single one of these proteins are expressed in male cells, but they just don't have this female-specific RNA scaffold that makes them into this antigenic pattern, antigenic form. So in summary then, I've told you that our journey studying with EXIST started with really basic understanding of epigenetics, dosage compensation, but now leading to this new model of EXIST in autoimmunity, and that is that this EXIST RNA is really a dominant source of autoantigens, which is sex-specific. In females, this leads to T and B cell chromatin changes. I told you earlier that maybe there are different genetic backgrounds are needed in this mouse model, and we think this is true in humans as well, because in a non-permissive genetic background, these chromatin changes happen, but it never progresses beyond that. But in this susceptible genetic background, this can go on to create autoantibodies, start leading to organ damage, and creating a cycle of damage and antigen release that we think can drive autoimmune disease. So this is an area of investigation that we hope can really further improve patient diagnosis and therapy. I want to turn our attention next to a second class of RNA, a shorter story, about circular RNAs and efforts to really think about them as new kinds of therapeutics. So circular RNA is another kind of really reasonably recognized class of RNA that arises from a process called back splicing. So many of you learn about messenger RNAs as these linear strings, but there's another form where the RNAs become a circle. And so if exons, which Dr. Mendell told you about, are spliced from 5' to 3', you get a linear RNA in order. But let's say, for example, exon 3 goes back to exon 1, you get a circle instead. Okay, so now this is a covalent linkage. What's very interesting is that RNAs are destroyed in cells by enzymes chewing on ends. So messenger RNAs last for hours, so they're very fragile, but circular RNAs are incredibly stable because they don't have this weak spot, and they can actually last for weeks in the body. And more recently, people have found that they can be really the source of binding sites of RNA binding proteins or templates for translation. But very interestingly, molecular biologists actually first learned about circular RNAs back in the 1970s because they're the genomes of pathogens. Pathogens for humans, like hepatitis D virus, pathogens for plants and bacteria, something called viroids. And so this harkens back to the theme of immunity is always kind of connected to these questions. So some of the questions when we started this work has to do with whether there's a self versus foreign distinction for circular RNAs. Do the cells care where these RNAs come from? How can circular RNAs be programmed to exploit their property for durable protein production? And how might they be used then for therapy? So our first studies really show that the cells do care about circular RNA origin. There is a system of innate immunity based on circular RNAs, and that is that any circular RNA that's made inside the cell is marked by a chemical modification called N6-methyladenosine, or M6A. And this tells the cell that this is a self product, leave it alone. So it's bound then by some RNA binding proteins that prevents it from activating this immune sensor called RIG-I. In contrast, if you make a circular RNA outside the cell that lacks this chemical modification, it will activate RIG-I, trigger a very strong basic interferon response and antiviral gene expression. And this information then gave us the ability then to create circular RNAs that's sensed either as self or as foreign. We can tune the immunogenicity actually up and down 10,000-fold using this information. And so if you want to use the circular RNA, for example, as an adjuvant for vaccination, you leave out the M6A. If you want to use it for gene replacement, gene protein production, you actually add the M6A, and the cell will treat it as self. A second aspect that we had to realize, work through, was how to get circular RNAs translated. So RNAs, you may know that messenger RNA translation happens by recognition of this chemical structure on the 5' end of the message called the cap. And this is how the ribosome to start from here and go forward. And you can see that circular RNAs have no ends, so how would they know where to start the tape? And so what was needed is an element, an RNA element called the internal ribosome entry site, or IRIS, which would then drive gene expression. So to find iris elements, a very talented postdoc, C.K. Chang, he developed a system where we can actually engineer reporter genes, this permuted in order, but then to get translated, they would need basically an iris element, and we could insert a large library of oligos, 55,000 of these, and screen for elements that can confer gene expression by the presence of ring cells. We identify a family of elements which then basically has iris activity, and we actually learn some of the rules that confer this class of activity to circular RNAs. This kind of idea we took forward to systematically identify pieces of RNA elements that are necessary for efficient gene expression. And this led to ultimately to the ability to make circular RNAs that have very strong gene expression output compared even to optimized messenger RNAs. Okay, and this is done by systematically screening different versions of pieces of the gene, 5' UTR, the iris, the 3' UTR, assembling them and screening basically by genome recombination, expression, and testing. Okay, and so to dig into the details, what we learned, there are really two lessons. So first is that there are a series of elements that can be optimized, and these are some of the sequential improvements that we can make. And the second point is that these improvements are modular. They stack on top of each other. So a series of 2 to 3 or maybe 5-fold improvements, you add it together, you can actually get to more than 200-fold improvement in circular RNA translation. A very important aspect of RNA therapeutics is RNA delivery. And here, working with our Stanford chemists, Paul Wender and Bob Weymouth, we use a class of compounds called CARTs that will complex with the RNA, deliver them into cells, but then let go of the cargo so they wouldn't get in the way of translation. Now you can see that these chemicals allow delivery through a number of routes for in vivo gene expression or RNA expression. So using these approaches, we can then test our RNA cargo, here expressing human EPO in mice. So EPO, of course, is the gene encoding erythropoietin that boosts red blood cell count. This would be something that's not allowed in the Olympics, but it's very important for patients with anemia, for example. Okay, so if you give EPO through a messenger RNA, it's expressed, but of course by 48 hours, it's all gone because the RNA is unstable. Whereas if you give it with the circular RNA, the human EPO can be detected in mouse blood for actually almost 2 weeks. And so importantly, on the right-hand side, now we're looking at the reticulocyte count. This is the pharmacological effect of EPO. And of course, the mRNA is too short, no effect, but circRNA can actually boost reticulocyte count with just a single delivery. So this is a technology now we're also very interested in. Let me just show you that we can also redose in vivo. So if you give a delivery, this is now expressing a nanoluciferase. These are mice. Now looking at the ventral view, this is a delivery that goes mostly to the spleen. So that's why it's on the right-hand side. And you can see that you get expression for 2 weeks and you can redose and you go back up again. You can keep going. So we're also interested in taking that technology to using for example, immunotherapy and vaccine. So this is the same idea, different optimized elements, but now driving an antigen, in this case the model antigen check over albumin, no M6A for immune activation. We can exploit the ability of this RNA to act as an adjuvant, so both the antigen expression and adjuvant in the same molecule for strong T cell response and activating immune activation in the draining lymph nodes. And in this, and you can then see in this vaccination experiment, very strong T cell responses both in the spleen and in the resident tissue in the lung, compared to, for example, peptide vaccination, which is the prior workhorse. And in this model of cancer eradication, we inoculate with melanoma cells expressing OVA antigen, but then treat with circular RNA OVA vaccine, and then we see a strong reduction of tumor burden And in fact, we think that this is mostly residual volumes from immune cells, and in fact, the reporter gene expression confers eradication of tumor cells. And this is, I think, then one of the really exciting directions that both mRNA vaccines and actually perhaps circular RNA-based therapeutics also in the field of immunotherapy. So in summary, I've told you about sort of two lines of investigation: exists RNA protein complex as a dominant female-specific driver of autoimmunity, and secondly, circular RNAs as a new family of pervasive stable RNAs. We can use genomic technologies to enable discovery of functional circular RNA elements, and that engineering these RNAs could improve protein production and perhaps vaccination in vivo. I want to thank many individuals I have the good fortune to work with, many generations of trainees, many colleagues, collaborators, and I want to highlight Will Greenleaf for his contribution in the invention of ATAC-seq, widely used throughout the world nowadays, my funding sources, and please know my disclosures. Thank you so much for your attention. Problematic is not the exist inside the nucleus doing its normal job. What is problematic is the exist RNA protein complex that's coming out of dead and dying cells. That is the stuff that you have to clear out. And so our sort of research now is directed towards strategies that might be able to detect cell-free exist RNA protein complex and how to manage that. Your second question is about circular RNA and whether that's the bridge to CRISPR. That's certainly a possible sort of strategy, and I think that even in the field of delivering CRISPR, which is obviously a way of doing gene editing, people are using basically all RNA components, right? So basically mRNA encoding Cas9 and the sgRNA, the guide RNA, for directing where the CRISPR goes. There are reasons to think that a circular RNA encoding Cas9 would actually be more efficacious because then the RNP would last longer in the intended tissue to raise your efficiency. But of course, beyond gene editing or maybe gene re-expression, there are other reasons why you might want to have a therapeutic intervention that is basically you can tune based on the time that you want rather than either permanent or none at all.
Thank you very much. I enjoyed the talk. It's quite interesting to see how Professor Mendell was listening to the role of circulating RNA while advocating the amino-associated viruses. So I want to ask, would you think for the local effect of gene therapy, DNA-associated viruses remain better than using RNAs or circulating RNA in that respect, especially after healing?
Well, I think after listening to Dr. Chang's presentation, that I was very impressed with the potential for translation. It was very apparent, and the field he is in, I think, especially offers opportunities dealing with cancer. I think that is the real potential first, I would say the first applied principle especially. And as far as CRISPR goes and its application, CRISPR has had its trials and tribulations up to this point, especially in treating childhood disease. So I think, again, the application of CRISPR and using RNA is much greater in cancer than it is in single gene disorders at the present. But I believe in CRISPR. I believe in the potential for CRISPR for all the reasons I said that we feared and had concerns about gene insertion. It's not the place to start. I think we have to have a stepwise approach in single gene disorders, and that has been, I guess, something that I have learned over the years that take your time, but on the one hand, the researchers like me want to go step by step. If you're out there and you have a child who has a lethal disease that will can only be turned around in months, you want it to go as fast as possible. So we, in a sense, those of us who know the safety issues and how the advances in the stepwise approach are more likely to lead to success, have to, or excess. Success is actually a good word to use in talking to families with these diseases. But I think Howard is really on the right path for treating cancer.
Thank you.
Do you think now, as we diagnose these disorders prenatally, do you think there will be a role of earlier intervention, and whether actually early intervention on the, for example, the muscular dystrophy syndromes would have a longer duration of effect compared to somebody who's had the disease for some till the age of 2 or 3 or 4?
Well, that's the question about duration. And so far, I think that if we can treat the disease as early as possible, we have much better and much greater opportunities for reversing the disease. So thinking about what you said and trying to do a prenatal gene therapy gives us the greatest opportunity without question. But as much as I like to be the bold pioneer, I don't think I want to be the first one to do that.
Go ahead. I think we have a couple questions from the doctors here, Regina. We have the second row and the first row, and then everyone else right after that.
Thank you for explaining talk. My question is to Dr. Chang. So one of the 3 proteins that interact with ADP is WTAP. And we know that WTAP is required in M6A because you are studying it. And you also know that M6A is involved in epigenetic modulation. Have you looked at the pH role in X chromosome silencing, especially considering that it is in the ADP?
Yes. Yes, indeed. We definitely noticed that WTAP, which is a component of the writer enzyme that deposits the M6A, is part of what interacts with the A repeat. And I think the work actually of Mitch Gutman and Sammy Jaffrey indicated that exist RNA is highly M6A modified, and they believe that there's a role for M6A in helping either stabilizing exist or in gene silencing. And so, I think but nonetheless, all the studies indicate that SPEN is probably perhaps the most important initiating factor in the A repeat. And the role of the M6A modification could either be in stabilizing that structure to make sure that it forms correctly and to attract SPEN, and or stabilizing other parts of the RNA, which we also have some evidence of that there's communication between different parts of the exist domains.
So thank you, Dr. Mendell. So, this lecture would not go without me asking a question for you. So as a pediatric neuromuscular neurologist between you, I feel like we all feel grateful in the neuromuscular community about what you did. And I just recently had a baby whom I dosed with Zolgensma almost 6 weeks ago, whose brother is debilitated in a wheelchair. And then the family finally has a hope of a healthy child. So thank you for that. So the question that I have for you specifically, where do you see the future of the vector use of AAV? I know that you've done work on plasmapheresis to circumvent pre-existing immunity or redosing. Where do you see the future of the vector use in the gene therapy world? Do you see it's more AAV? Do you see lipid nanoparticles? Where do you think this is heading in terms of the vector's usage?
Thank you for the question. Let me respond first by saying I didn't clarify. I mentioned about pre-existing immunity, but didn't clarify. I was thinking about, whenever you present it, you think about what did you leave out. And what I didn't say was that the pre-existing immunity comes from natural exposure. These adeno-associated viruses are everywhere, and only a certain percent get antibody buildup to that. It may have something to do with X or Y chromosomes, and I think, but it's about 15% of the community will develop immunity to adeno-associated virus, at least the ones we've used. So now you have a natural barrier for treating patients. That's the first question. The second question is a more challenging one. That is, once you give the virus, can you then re-administer the virus at a later time if the gene effect over time is lost? So my hope is that we start with pre-existing immunity because the viral titers are generally lower. And I've been pushing very hard to get that accomplished. Pushing means that we do studies we've learned and clinical approaches that we've learned for 40, 50 years using plasmapheresis for autoimmune disease, and it's certainly very effective. And that's the first approach. And that study is being launched now by Sarepta. So we'll have the answer to that within, I would say, a reasonable amount of time, whether we can lower pre-existing immunity well enough and safe enough to re-administer the virus. My personal view is that we can. We did it in non-human primates. So with pre-existing immunity to AAV8 in non-human primates, we were able to administer the virus successfully. Now the challenge will be if we have viral titers that are 100,000, 200,000 dilution as opposed to 1 to 400, 1 to 800 in pre-existing immunity. Can we then re-administer the virus without immune suppression? There are very effective approaches that are being developed using monoclonal antibodies, and I think that's one approach. It hasn't been tested clinically. I'm not a real excited fan about using sirolimus or tacrolimus as opposed to plasmapheresis. So, but we may end up doing that as well, or having chemotherapy that is in combination with. And then there are the other approach, I think, which will be done. You know, you can think things are impossible today, but 20 years from now they won't be, and that is developing serotypes that are tolerable that won't cross-react with AAV-RH74 or AV9. So I think we have a variety of approaches, and I think in the end it'll all be successful, but it may be a little painful getting there to the answer.
Thank you.